Welcome to all. I am Dr. Asha Kadam from Department of Botany, Dada Patil Mahavidyalaya, Karjat. Today we learn about the plant constituent, that is the endoplasmic reticulum. In the endoplasmic reticulum, we learn about the morphology of endoplasmic reticulum, functions of endoplasmic reticulum, ultrastructure of endoplasmic reticulum, and also the chemical constituent which are present in the endoplasmic reticulum. So first we learn the introductory part of the endoplasmic reticulum. We know that in 1945 the lace-like membrane of the endoplasmic reticulum was first seen in the cytoplasm of chick embryo cell. These are the membrane bound structure, or we also call as the membrane bound channel, that's seen in the form of a network of delicate strands and also the vesicles in the cytoplasm. Means in 1945 the lace-like structures are present in the cytoplasm. They coined the term, that is the endoplasmic reticulum. These endoplasmic reticulum also having the different elements like the vesicles and the different delicate strands, or we also call as the different network of framework. When we learn about the location of the endoplasmic reticulum, that time we know that these endoplasmic reticulum that are mostly found in the eukaryotic cell. These are found to absent in the mature erythrocyte, ova and the embryonic cells, and also the prokaryotes. Means the endoplasmic reticulum are present in only the eukaryotic cell, but there are some exceptional cases like the mature erythrocyte. In the mature erythrocyte, endoplasmic reticulum is completely absent. Also in the ova and the embryonic cells. In the ova and the embryonic cells, endoplasmic reticulum is absent. Means exceptional of these mature erythrocytes, ova and the embryonic cells, remaining in all the eukaryotic cells, endoplasmic reticulum is present. In all the prokaryotic cells, endoplasmic reticulum is absent. Means endoplasmic reticulum in the prokaryote is always or totally absent. So when we learn about the amount of endoplasmic reticulum, that time we know that the endoplasmic reticulum varies in amount of cell to cell. Endoplasmic reticulum forms 30 to 60% of total membrane in the cell. Means when we consider the total membrane of the cell, that time we say that approximately 30 to 60% endoplasmic reticulum occupies the space in the cell. Now, how this endoplasmic reticulum is originated? At present manner, the origin of endoplasmic reticulum is not clearly known, but the most concrete hypothesis about the origin of endoplasmic reticulum, that is, it is the budded of the nuclear envelope. Means it is the bud from the endoplasmic the nuclear envelope. So the most concrete hypothesis regarding the origin of endoplasmic reticulum is this endoplasmic reticulum is budded off from the nuclear envelope. The endoplasmic reticulum that appears to arise from the outer membrane of the nuclear envelope by the inner membrane of the plasma membrane. So, these endoplasmic reticulum are located outer membrane of the nuclear envelope to the inner membrane of the plasma membrane. We know that these two organelles, that is the nuclear envelope and the plasma membrane, they are outer and the inner membrane, means they are double membrane bound structures, and these endoplasmic reticulum located from the outer membrane of the nuclear envelope to the inner membrane of the plasma membrane. So, the smooth endoplasmic reticulum that are arise from the rough endoplasmic reticulum by means of the detachment of the ribosomes. We know that on the framework of these endoplasmic reticulum, there are presence of the ribosomes. When the ribosomes are present, that time we call as the rough endoplasmic reticulum. And when from these rough endoplasmic reticulum, there is the detachment of the ribosomes, that time they are forming the smooth endoplasmic reticulum. Means these smooth endoplasmic reticulum are originated from the rough endoplasmic reticulum by the detachment of the ribosomes. On the basis of morphology, endoplasmic reticulum having two types, that is the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Smooth endoplasmic reticulum we also call as the SER, and rough endoplasmic reticulum we call as the RER. The endoplasmic reticulum membrane is much thinner as compared to the plasma membrane. Means the membrane of the endoplasmic reticulum approximately having a thickness that is the 50 angstrom unit, whereas the thickness of the plasma membrane is the 80 to 100 angstrom thickness. Means when we compare to the another organelle to this endoplasmic reticulum, that time endoplasmic reticulum having a much more thinner or much more very thin surface or the membrane than the other organelles. We take the example of a plasma membrane. These plasma membrane having a thickness that is the approximately 80 to 100 angstrom, whereas in the endoplasmic reticulum the membrane having a thickness that is the only 50 angstrom. Hence we say that endoplasmic reticulum is much thinner than that of the plasma membrane. So here it is a diagram showing this diagram which showing the ultrastructure of endoplasmic reticulum. So on the basis of morphology, where the ribosomes are absent, that time the appearance of this network or framework is very smooth and soft, and that time we call as the smooth endoplasmic reticulum. When this framework or the network of the endoplasmic reticulum having a ribosomes on their surface, that time this framework of endoplasmic reticulum having a appearance which is too rough, and that's why this roughness this endoplasmic reticulum called as the rough endoplasmic reticulum. So these are the small and the tiny particles that we called as the ribosomes. These are the flattened sac-like structures which are unbranched, that's called as the cisternae. These are the nuclear envelope. Nuclear envelope having inner and the outer membrane. So, this one is the nuclear pore. This nuclear pore and the nuclear envelope are the elements of the nucleus. We know that nucleus is the central core part of the cell which controls all the activities of the cell. So, this endoplasmic reticulum is located from the outer envelope. Look this, here is the outer membrane of the nuclear envelope, means this endoplasmic reticulum or the network of this endoplasmic reticulum is located from the outer membrane of this nuclear envelope to the inner membrane of the plasma membrane. So, this diagram showing the ultrastructure of the endoplasmic reticulum. In the two cisternae, they are having an intermediate space, and that space we called as the cisternal space. That called as the cisternal space. Then we learn about the physical structure of endoplasmic reticulum. The endoplasmic reticulum is three-dimensional network of intracellular structure. It is formed main three elements, that is the cisternae, tubules and the vesicle. Means when we learn about the physical structure of endoplasmic reticulum, that time we say that endoplasmic reticulum is a three-dimensional network on which there are the three main elements are present, that is the cisternae, tubules and the vesicles. So, first we learn about the cisternae. These are the flattened, unbranched and sac-like elements. Cisternae are flattened, unbranched and the sac-like elements. They always lie in stacks parallel to one another. They bear the ribosomes on the surface of these, and that's why the appearance of these cisternae is very rough. These cisternae also contains the enzyme, that is glycoproteins. This contains the proteins, or we said the glycoproteins. These glycoproteins having a name, that is the ribofourin 1 and the ribofourin 2 that bind to the ribosomes. These cisternae contains the different types of enzymes and the proteins. For example, when we consider about the protein, that time here the glycoproteins are always present on the cisternae, or these cisternae having involvement of some glycoproteins like the ribofourin 1 and the ribofourin 2, which always binds to the ribosomes. So, next elements of the endoplasmic reticulum is tubules. We know that tubules are the irregular branching elements. Means these tubules are the irregular branched structures which form the network along with the other elements. These are often free from the ribosomes. Means tubules are without ribosomes. They are the irregular and the branching structure of endoplasmic reticulum. And the next element of the endoplasmic reticulum, that is the vesicles. So, these vesicle name itself indicates its meaning. Vesicles are oval, rounded and vacuole-like structures, or we say that these are the elements. These also free from the ribosomes, means when we consider the three elements of endoplasmic reticulum, that is the cisternae, tubules and the vesicles, that time we learn that cisternae having the ribosomes or cisternae with the ribosomes, which having a rough appearance, presence of a different enzymes and proteins. For example, glycoproteins like the riboflavin first and the riboflavin second. Then tubules. Tubules are the irregular branched structure of the endoplasmic reticulum, which are free from the ribosomes. And the last element is the vesicles. These vesicles are oval, round-shaped and vacuole-like elements or the structure that are also free from the vacuoles. Means in short, cisternae having the ribosomes and tubules and the vesicles are free from ribosomes. So, all the elements of the endoplasmic reticulum, they are freely communicated with one another and also contains the fluid, and that time we call as the endoplasmic matrix. When the elements, they are communicated with each other via some fluid, and that fluid we call as the endoplasmic matrix, which are present in the lumen of endoplasmic reticulum. So, this matrix is different from the cytoplasmic matrix, which are outside the endoplasmic reticulum. Means the fluid-like matter which are present in the endoplasmic reticulum lumen, that we call as the endoplasmic matrix. And this endoplasmic matrix is always different from the cytoplasmic matrix. So, the endoplasmic reticulum, they may pass from one cell to another through the plasmodesmata in the form of a desmotubules. When the endoplasmic reticulum, they are passed from one cell to another, that time they are passing through the plasmodesmata in the form of a desmotubules. So, in this diagram, these are the two cells. Through these two cells are communicated, or when they are passing some material, that time these material are passing through the special structure that called as the plasmodesmata or the plasmodesma. Through these tubule-like structure that we call as the desmotubules, means here the neighboring cells are communicated or linking their protoplasm through the plasmodesma by desmotubules. So, these are diagrams showing the ultrastructure of endoplasmic reticulum, on which we know that this endoplasmic reticulum is framework or network-like structures which are located from the inner membrane of plasma membrane to the outer membrane of nuclear envelope. This one is the part of the nucleus, in which there are the nuclear pores are present. These are the double membrane structures that we call as the nuclear envelope. Here, these are the tubules and the vesicles, which is the elements of the endoplasmic reticulum that are free from the ribosomes, and that's why they called as the smooth endoplasmic reticulum. Because of absence of the ribosomes, the appearance of this framework or the network is very soft and smooth, and that's why this endoplasmic reticulum is called as the smooth endoplasmic reticulum. Then this network having some ribosomes or some tiny particles that we called as the ribosomes. And here these are the flat sac-like structures which are unbranched that we called as the cisternae. These cisternae are lies parallelly to each other. And we know that cisternae also on their membrane having the ribosomes. And due to presence of the ribosomes, this network or the framework of the endoplasmic reticulum is having a appearance rough, and that's why it is called as the rough endoplasmic reticulum. We know that rough endoplasmic reticulum there is included these cisternae which are with the ribosomes, which contains the different enzymes and the proteins. For example, glycoproteins. Okay. So, next to this, we learn the molecular structure of endoplasmic reticulum. We know that the membrane of endoplasmic reticulum that are composed of two layers of a phospholipids. The membrane of endoplasmic reticulum that are composed of two layers of a phospholipid molecules which are sandwiched by two layers of a protein molecules. Means here the membrane of the endoplasmic reticulum are sandwiched in between the two layers of a phospholipid layer and the two layers of a protein layer, or we also called as the some are the phospholipid molecules and some are the protein molecules. They are having a different layers. They are sandwiched by each other. Okay. So, like the cell wall, in the cell wall also there are some phospholipids molecules and the proteins are sandwiched by one another. Here also the membrane of the endoplasmic reticulum that are composed of two layers of a phospholipid molecules which are sandwiched by the two layers of a protein molecules like the other membrane of the cell wall. Next to this, that is the types of endoplasmic reticulum. We know that on the basis of a morphology or where there is the presence of a ribosomes or absence of a ribosomes means the network of a endoplasmic reticulum with the ribosomes or without the ribosomes, they are having a two types, that is the smooth endoplasmic reticulum and the rough endoplasmic reticulum. So, when we learn the endoplasmic matrix, that time we know that the space inside the tubules and the vesicles that are filled with the fluid-like structure or the watery medium that is different from the cytosol to the outside of the endoplasmic reticulum, that we say that endoplasmic matrix. Means in short, endoplasmic matrix is nothing but the fluid-like matter which are present inside the tubules and the vesicles of endoplasmic reticulum, which are totally different from the cytoplasmic matrix. Their walls are always constructed of lipid bilayers membranes that contains the large amount of proteins which are similar to the cell membrane. Means the all of the endoplasmic reticulum that are constructed by the lipid bilayer membranes that contains the large amount of proteins which are similar to the cell membrane, or we say that the plasma membrane. So, next to this is the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is an arrangement of the tubules and the vesicles. The size and structure of this smooth endoplasmic reticulum always varies from cell to cell. It can change within a cell lifetime to allow the cell to adapt some changes in its functions also and in its various requirements. So, there are no ribosomes are present on these membrane surface, and that's why the appearance is very smooth. This smooth endoplasmic reticulum is connected to the nuclear envelope. The network of these smooth endoplasmic reticulum that allows there to be enough surface area of the action of storage of some key enzymes and the products of some enzymes. These are less stable as compared to the rough endoplasmic reticulum. Smooth endoplasmic reticulum is the characteristic of cells in which synthesis of non-protein substances are take place. Means the characteristic of the cell is that is the cells in which there are the synthesis of non-protein substances take place. Means always on the smooth endoplasmic reticulum membrane there is synthesis of a non-protein content molecules. Next one is the rough endoplasmic reticulum or RER. The surface of rough endoplasmic reticulum is studded or attached with the ribosomes, and due to attachment of these ribosomes, the appearance of this membrane surface is rough. So, it mainly consists of the cisternae. We know that cisternae having the ribosomes. The membrane of rough endoplasmic reticulum forms the large double membrane sheets, which is located near and continuous with the outer layer of a nuclear envelope. So, rough endoplasmic reticulum having a ribosomes, that's why they having a appearance very rough, and it included the element that is the cisternae, which is the with ribosomes. Means on the membrane of the cisternae, ribosomes are present. Next, rough endoplasmic reticulum also play important role in the synthesis and packaging of some proteins. For example, Russell's body of plasma, Nissel's granules of nerve cells. Binding site of ribosomes on the rough endoplasmic reticulum is the translocon. When we say that the binding site of the ribosomes on the rough endoplasmic reticulum, that is the translocon. So, the ribosomes are bound to this rough endoplasmic reticulum at any time. They are not stable part of the organelle structure because ribosomes are constantly being bound and reached the membranes, and these ribosomes that only bind to the rough endoplasmic reticulum once a specific nucleic acid complex is formed. This nucleic acid complex is formed in the cytosol. So, there is the special complex which forms when free ribosomes begins translating the messenger RNA of a protein which is destined for the secretory pathway. And so, the first 5 to 30 amino acids, they are polymerized, encode a single peptide and some molecules which are play important role in carrying some messages. which recognize and binding the single recognition particle. That single recognition particle is called as the SRP. That is called as the SRP. So, ribosomes, they are attached to the endoplasmic reticulum, synthesize all the transmembrane proteins and also secreted some proteins which always store in the Golgi apparatus, lysosomes and the endosomes. So, translation pauses anti-ribosome complex, they are bind to these rough endoplasmic reticulum translocon. So, they also play important role in the transport of a protein. As the proteins, they form in the endoplasmic reticulum which are transported through the tubules. They are always transported through the tubules towards the protein of the smooth endoplasmic reticulum, which lie nearest to the Golgi apparatus. Means there is the transportation of a proteins in the endoplasmic reticulum via the tubules, which lie nearest to the Golgi apparatus. And we know that tubules and the vesicles, these elements are included in the smooth endoplasmic reticulum. Means smooth endoplasmic reticulum is play an important role in the transportation of protein molecule and also the synthesis of non-protein content. So, at this point, the small transport vesicles that are composed small envelope of smooth endoplasmic reticulum which continuously break away and diffuse the lipase layer of the Golgi apparatus. So, inside these vesicles, there are synthesis of the endoplasmic reticulum. So, also they play an important role in the transportation of the vesicles. We know that they are surrounded by the coating protein. That coating protein we called as the COP first, COP second, that is the develop the complex that we called as the coat protein complex. That called as the coat protein complex. So, COP second always target the vesicles to the Golgi apparatus, and transparent proteins, they form the rough endoplasmic reticulum to the Golgi apparatus. So, this process is called as the Anterograde Transport. When these COP second target the vesicles to the Golgi apparatus, there is the transportation through the vesicles. So, these transparent proteins, they from the rough endoplasmic reticulum to the Golgi apparatus, and that time we called as the Anterograde Transport. This process is called as the Anterograde Transport. But when these COP first complex, they transport the proteins from the cis end of the Golgi apparatus back to the rough endoplasmic reticulum, that time these transport of a protein or these process of the transportation of a protein is called as the retrograde transport. It called as the retrograde transport. So, here this diagram showing the transportation of the vesicles via the Golgi apparatus. So, these are the nuclear, rough endoplasmic reticulum. There is the transportation of the proteins through these vesicles via the Golgi apparatus. Second method of transport of the endoplasmic reticulum that are involve the membrane contact site, where the membrane of endoplasmic reticulum that are organelles held together, allowing the transport of a lipid and also the small molecules.